Thank you very much and welcome after a lunch break. It is really a pleasure to bring today a very illustrious panel to deal with a topic that is very important in terms of advances in cancer therapeutics. I am Esteban Pombo. I am the Chief Technology Officer at TargImmune Therapeutics, trained as a medicinal chemistry, worked for Novartis and other companies, and have been with TargImmune Therapeutics for 6 years. We are getting very ready now to enter clinical studies with our novel nanoparticle technology for cancer treatment. My co-chair, Andreas Wallnoefer, will introduce the topic of the panel.
Yeah, a warm welcome also for me. I'm Andreas Wallnoefer. I'm a partner at Jeito Capital, which is a European equity fund based in Paris. Before that, I was for a long time at Roche. I was in the Roche R&D leadership team for more than 14 years, did different roles there, including heading the clinical research department, but also development, and interestingly enough, also cardiovascular metabolism. When we were preparing for this panel, of course, the topic is advances in cancer therapies. And we were debating, since many of the panelists are involved in companies in immuno-oncology, about the innovation cycles. I mean, we all have seen that, and that's not only in oncology. From my own experience, 10 years ago, all the big pharma companies wanted to leave cardiovascular metabolism, and see today, basically everybody wants to be in again. And these are the big runners. You see it in other areas happening. Think about rare diseases. Everybody wanted to be in rare diseases. Now everybody wants to get out of rare diseases. And in oncology, we had, of course, the big interest and dynamics in immuno-oncology. And today, some of that has cooled a little bit off, and everybody wants to do ADCs. Now, the question is, what will people want in a few years from now? And maybe the reality is, in fact, if you think about sustainable cancer treatments, that you have to combine ADCs with immuno-oncology. Immuno-oncology is probably the only way that you can achieve sustainable cancer therapy. So when we were debating all of this, we said, okay, let's give the whole debate a little bit of twist and talk about these cycles, what it means for our companies here on the panel, and then open it also to the broader audience, but also what it then means for the CEOs, the executives managing these companies, where you need to have some consistency. You cannot change direction every other year because somehow there is a new trend. And lastly, most of the most successful companies are the ones that really believed on their idea and had some continuity. So with that opening, I would just pick somebody, maybe Apollon. I mean, you are running an immuno-oncology company. Why you think this is a good idea?
Maybe start with the 2-minute presentation of who you are in your company before you proceed to the question.
Yeah, happy to do so. So, hi everybody, my name is Apollon Papadimitriou. I'm the CEO of IOmx Therapeutics. I'm a biochemist by training. I had a prior life. Prior to IOmx, I was on the dark side, with Big Pharma, a company called Roche, for more than 20 years, together with Andreas. And now I switched to the more bright side into biotech since 6 years, and I'm the CEO of IOmx. IOmx is a company that is strongly believing in immuno-oncology. We continue to believe into our story, and I will try to explain to you later why we believe in that. We are a clinical stage company. We are trying to identify novel immune escape mechanisms in the tumor microenvironment. And then we want to drug these targets that we identify by, very potent drugs that have the chance to drive single agent efficacy. And that also brings me to trying to answer the question that Andreas started off with. I think, of course, as you were saying, you see ups and downs, you see trends. And as a small company, you of course have to be flexible, but you cannot follow every trend, right? But what you can do is to do a very robust analysis and ask the question, why have others not been successful? Why is, the story coming down to a consolidation, and that's what we are seeing. And then you can ask the question, how are we different, and how do we fit into this, consolidation? And this is exactly what we have done from the get-go in our company. And just to mention one example, the whole, immune checkpoint field was driven. I think we understand it's a wonderful story, right? PD-1 is a wonderful product. The antibodies against it. It's a nice target. It's a nice biology. It has revolutionized the way how we are treating cancer. But what's next? And then you saw the second generation products coming as add-ons, trying to combine everything on top of PD-1, typically lacking a strong single agent efficacy. And maybe that's part of the problem, right? So you were seeing nice signals in phase 1Bs. People were excited in combination. And then you had to do a true phase 2 type study, head-to-head comparison. And then people were shocked because the historic PD-1 data were showing more efficacy than the combination. So maybe that's part of the problem. So I feel, you have a chance if you are really committed to drive single agent efficacy, or you have a regulatory path to approval to avoid, this, head-to-head comparison studies. I can speak more about it in a minute.
So Apollon, your point is that 2 points. Actually, you need to have consistency to follow your vision and a critical view to learn what's happening around you to make yourself fit, differentiated, and effective in that setting. Yeah? But why don't we ask Eric? Eric, you are in the business for quite some time. Maybe if you quickly introduce yourself and how was your journey and how do you see this debate?
Okay, thank you. So I'm Eric Halioua, the CEO of Pharma, Pharma, company in cancer vaccine. So I'm just some background on myself. I'm molecular biologist and MBA, and I've been in the biotech for 25 years. I've co-founded several of them, sold one of them to Genzyme in 2002. All of them in cell therapy, by the way. And 8 years now with Pharma, which is a company involved in cancer vaccine. So we are using a unique and differentiated cell line that we load with GMP peptide to address different kind of tumor. And we are in clinical trial in non-small cell lung cancer currently, and we should have the final result of the trial second half of this year. So I think the point is interesting. It's 8 years now with Pharma. We raised 61 million euros in the company. We're a team of 42 people. But it has been, as I said, and to sustain the divisions, it's something which is very challenging in this area. When we talk about cancer vaccine, 10 years ago was when Merck, GSK, had a big failure on the phase 3. I don't know if you remember that. So it was before the checkpoint inhibitor approval. So it was one of the first generation, if we can call it first generation of vaccine, but it was a big failure. And 1 or 2 years later, we established a company to develop the program. And at the top of that, it's a cell therapy. So you can imagine you are not in the ideal situation, but we did it. We just need to believe on the product, to have the right data, and to be innovative in terms of trying to get financing, at least in the beginning. We get first alliances, a deal in Asia with LG Chem. In 2019. So it was before starting the clinical trial in the lung, and they were very interested by the approach. And based on this deal, we get tractions from Asian investor, and we get traction from KIP, Core Investment Partner, which is a big investor, 4 billions, and they came in consortium to finance the company with the Belgium investor, and the story move on. So you see, it's a different approach we have been following, but once again, 8 years later, we are still here. We have clinical data, and we are preparing now next phase 2B randomized study to demonstrate the clinical sign of efficacy we are demonstrating now. So I don't want to spend too much time on that, but I think continuity on the visions and to believe on the product and to stay optimistic and positive is definitely a key in this field. That's what I would like to say.
But Eric, you add another interesting aspect. If I interpret your story, you were also creative by finding new funding and finance partner if you went to Asia and got investors there, right? So essentially, it's a challenge not only on the scientific medical vision, it's also about agility in dealing with the investor community, right?
You're talking to me, huh?
Yeah.
Is what
No,
I said.
you are on.
Yeah. Yeah. No. As I said, in terms of financing, we have Asian investor, plus non-dilutive money from because we are based in Belgium. So almost half of the funding we get came from this non-dilutive money and really help the company and to be up to this stage. So it was for the situation of PDC line the way we find to finance the company. So to finance company is always key and to reach a value inflection point and to pass through this cycle, of technology impression or mode. Now, cancer vaccine is a lot more interest in the scope of pharma company, and I'm very pleased on that. And I hope it will stay for a long time. That's what I would like to say.
Excellent, Eric. I mean, and noted, and I take now the Jeito view. I think what is important is that you also have investors who provide you a certain stability and believe in your vision to give some continuity, right? So, but let's. I'm at random here, but let's ask Esteban. Your field is very interesting, and there is a lot of attention. So how do you see this situation where actually one day you may be in the center of attention, and then somebody has a negative trial, and you have to explain what that means for your company, right?
I think my mic.
Try again.
Does it work this time?
Good.
Yeah, there's not much hope for us if we can't figure out how the microphone works. So yeah, it's, thanks for the question. So it's a really pleasure to be on the panel and partake in the discussion today. So I'm Esteban Myatt, I'm CEO at Macomics. We're a UK biotech company spun out of the University of Edinburgh 4 years ago. Our focus is on macrophage drug discovery. So really at the heart of the company is a technology platform we call Enigmac, which allows us to identify and genetically validated novel biology for targeting macrophages. The platform is disease and actually modality agnostic. It relies on novel methods for genome editing iPS cells that we've developed inside the company. And this allows us, for the first time, and we think it's really a novel approach, and it's probably a world first, to run CRISPR-I and CRISPR-A, for example, screens, target validation programs in physiologically relevant human macrophage models, which has been one of the really big and significant challenges in the field historically. So that's a technology platform that's kind of enabled our pipeline. From a product perspective, our lead program's in IND studies. This is a first-in-class ligand-independent pan-LILRB antibody. We have a first-in-mechanism approach for targeting this receptor family. And we believe this receptor family is actually really important in immune suppression in tumor microenvironments, not only through macrophage immune suppression, but also in terms of T-cell immune suppression and NK cells as well. So it has a multi-hit kind of therapeutic hypothesis. For this program, we've identified a blood-based biomarker. We think in immuno-oncology, biomarkers are very, very challenging, but biomarker can also add considerable value when you're thinking about your clinical development path. So this is something we've invested in quite heavily. We've also used the platform to try and build out the pipeline in the company, focusing on first-in-class opportunities for targeting macrophages. We have also used it as a way of, in a way, creating options for the company, which I think is relevant to the question that's being asked. So last year, we entered into our first pharma partnership with a Japanese pharma called Wallnoefer. Ono was actually the first company globally anywhere to launch a PD-1 or PD-L1 agent. And this was on a target that we'd identified and validated in immuno-oncology that had arisen through our platform. So that's created another sort of, in addition to the equity that investment that we've raised, it's also brought in sort of non-dilutive financing through BD activities. And then we continue to leverage the platform and build our target portfolio beyond the lead program. So I think that my kind of view on this is that I think there is inevitably in almost all fields negative data sets. I think there's negative, and it is cyclical. There's been negative data sets in the ADC space, the kinase inhibitor space, the IO space. and in the macrophage space too. And I think our view on that is that each product is individual. You have to believe in it. You have to follow the data. You have to have continuity of kind of vision and purpose, but also be reactive and be able to understand those data sets that are emerging elsewhere. And a great example of that in the macrophage space, which I'm sure many of you are actually familiar with, it's been constant news flow of this over the last 12 months or so. It's been the challenges in the CD47 SERP alpha axis, right? And this using this approach to induce macrophage phagocytosis in a blood cancer context. And actually, I think what this really actually tells us in this specific instance is that it's really important to match the therapeutic context and the disease biology to the therapeutic mechanism, because there's a lot of macrophage heterogeneity. They're very plastic cells. They're involved in all sorts of different biological processes: antigen presentation, cytokine release, phagocytosis, efferocytosis, synaptic pruning, astrocyte regulation. So it's extremely broad. And the really key thing is to understand the fundamentals of the biology to match the right mechanism to the right disease context. And I think it is unfortunate that sometimes, negative news flow like that, in general, people will say, yeah, but the macrophage space is not playing out because of what's happening on CD47. That's actually a very specific approach. And I think, you have to really start from the fundamentals of the biology and build your clinical rationale from there. But it's all about creating options.
Excellent, Stephen. I mean, you added another important dimension to the state debate. It's in a way it's obvious, but not necessarily obvious, which is essentially you need to focus on the content, on the science, really understanding what you are doing. That allows you, to Apollon's point, to differentiate, to explain how you put your programming context of the environment, to really have a deep understanding of the biology, the mechanisms, and actually also to communicate this to your investors and hopefully find investors who actually embark on the same journey to be aligned to finance you through.
And I think what's really important, and I completely agree, and I think what's really important in that approach is you don't exist in a silo, right? As an organization, you have your investors who hopefully are aligned with the vision for the company and where you're going. But also you need to be, I think it helps to be very externally orientated as well, and be continuously seeking that kind of market feedback and understanding from prospective pharma partners, be that, post the pivotal trial of your drug or for a target discovery collaboration. What is it those partners need to see to transact on that program? And I think that's absolutely critical then when it comes back to taking that market information back into those investor discussions, so that you have a very clear understanding of what you actually need to deliver to get to that value point from a partnership perspective. I think it's obvious again, but it's sometimes forgotten that we don't exist, in a silo.
Excellent. Why don't we ask Aram, right? So particularly also in the context of the latter statement, right? So what you see at the beginning of a hype, you can create a lot of enthusiasm with little data. Then particularly if you're on the top or on the down of the hype, then you really need to go deeper, and your partners, investors, pharma, everybody gets much more critical, and your package needs to get more and more solid. Now, tumor microenvironment is, of course, a key target for many approaches, and your company is particularly focused in that area. So, what is your experience in that sense?
Thanks. Well, let me just briefly introduce the company. So TME Pharma, we're a Berlin-based biotech. We use RNA aptamers to modulate the tumor microenvironment, and we're focused on solid tumors, in particular glioblastoma right now. And I think this Andreas goes really to the core of your question. It's an indication that I think the board of the company and the investors of the company tried to avoid. But they ended up pushing the company toward indications that the technology wasn't going to be optimal in. So we finally got to glioblastoma, and there you're going to learn obviously from the preclinical work where we saw 100% complete response in spontaneous tumor models. But we got to the clinic, and of course, we combined our drug with radiation therapy, and it didn't quite turn out that way. We saw high rates of response when we combined our inhibitor NOX812, which works on a chemokine, very important for the repair of blood vessels by vasculogenesis, but also for immune privilege of tumors. And we had to learn in the context of the trial. So we're in the phase 1/2, and we were lucky to be working with a very good translational group at the University of Bonn, Michael Holzel's group, and he did two really important things. And it's something that I think you have to be willing to do, is to change your mind in these trials. He found a biomarker, which we're not probably going to use, but it really validates the mechanism of action, because the patients who responded to our drug as monotherapy were the ones that had the highest amount of target in the blood vessels of the tumor and on the tumor cells themselves. So that gave us confidence that our drug was actually doing something biologically important. The other thing he identified was that, contrary to the animal models, the other way of rebuilding vessels, angiogenesis as opposed to vasculogenesis, was actually very important. It was turning up in other parts of the tumor where our target wasn't. And so we made the decision to actually combine with an anti-VEGF in addition to an anti-CXCL12. And that's where we saw really some exceptional data. So 100% radiographic response, 83% MRANO response, which was durable. We saw half the patients get to either complete or near complete responses. It's a small expansion group, but we have a really exceptional signal. And based on a matched comparator cohort who was getting standard of care, we doubled the median OS. And more dramatically, when you look out 18 or 21 months after start of therapy, we're 10-fold higher in survival than standard of care. So it's an exceptionally strong survival signal, but it took us a long time to figure out how to use this drug to find the right combination. And it really demanded digging into the data and figuring out and working with investors to convince them that, look, this isn't exactly what we hope, but there's a really good reason that the next piece should turn out better.
So there's a lot of understanding of biology and how the biology evolves from the early creation of the project. And you need to keep getting data and trying to solve problems and challenges, because really getting to that very impressive results in glioblastoma is a great success. But getting there was very difficult, and it took a long time from the original inception.
Yeah, it did indeed. And I think raising the money we needed to get there was extremely difficult on the public markets.
I think that connects well with, I think Stephen gave the statement that you need to understand what pharma, what investors are looking for, right? At the same time, in my experience, big pharma was some of the biggest failures we had there was when we tried to twist the product to marketing needs the product could not deliver, right? So there is a fine line not to be ignoring the demands while staying with your story, right? And so maybe to conclude the round, Konstantin, you are a Secarna that's again in immuno-oncology, but you have a different approach there. So what is your learning here?
Yeah, thanks. So we are a little bit like Steven, we're a platform technology company. So basically our core expertise initially is not immuno-oncology, but really it's the field of antisense, oligonucleotide technologies. And this is where, similar to Macomics, actually utilize the platform to deliver or to partner up with pharma and biotech to actually be the discovery engine on their targets. And of course, utilizing those funds, those revenues to also build up slowly, step by step, selected proprietary programs. And actually, I think why we are in immuno-oncology with the most of our proprietaries is
a little bit like serendipity, let's put it like that way, because just some of those targets were really interesting to use the modality, because antisense is a modality that may not be fit for all indications, for all targets, and maybe for some of those targets there might be a more clever solution. Like if it's a receptor, you might think of an antibody as the more clever approach. But I think in our case, in the most advanced product we do develop internally is an immuno-oncology actually receptor. And there's good reasons to believe why an antisense in that case might be the more clever approach. And this comes back to the points we've heard. I think it's a combination of, at the end of the day, it's data, and you have to understand not only the target data, but also the tech data. And we also had those waves, and I mean, the antisense it's not completely new, right? It's been there for decades, but the first generations were just not good enough. You get knockdown of the target, but you don't have drug product-like characteristics, which is, I think, something many startup and biotechs initially don't consider, that you just don't need to see, or it's not enough to see effects in a cell line, but you need to make up your minds, how do you actually, at the end of the day, get a drug product out of it? And I think this, in our case here, at the end of the day, we target a receptor called neuropilin-1, which also is not completely new, but there's more and more understanding. So also to the point, we need to always have in mind what is the competitive environment, what are new data that actually drives the target to a certain indication, what enables us to better understand maybe certain biomarkers that either speak for targeting such a program or actually also deleting it. So really, I think hanging on to some targets, and then many have done, and maybe we also have done in the past, you have to, at a certain point of time, really rather stop quickly and decide on progress only a few and get focused, instead of being as a small company on 10 different programs. And so maybe back to your question, on our hand, we really did a deep dive into, is it the right target for the modality? Do we actually understand our technology? That's for sure. But do we understand the target good enough to proceed? And of course, all the way up to today, and we're going towards pre-IND, we believe that the data that we see is convincing and good enough and competitive. But of course, we can't stop there. And you always have to consider also what's new in the environment to say, what else do we actually need to dive into, what else data do we need to show to also keep up the discussions with the investors and also with the farmers, because at the end of the day, you mentioned it, they always want to see more. So it's on us to understand what do they need and actually how can we prove that.
Excellent. So what we now want to do is actually also include you into the discussion to share from your perspective as entrepreneurs this dynamic of innovation cycle, opportunity, challenge, and how do you deal with it. But before I do that, let me try to synthesize what I think we come out as an aligned message here, right? So I think what you hear from all the panelists is the importance of having a clear vision and conviction about what you want to achieve with your approach. There was a very clear vote, I think Stephen said that, about following the science. You need to really have a deep understanding how you're going to go to interfere with the pathology. There was Apollon's statement that basically only that allows you then to differentiate and understand the field and how you position. Konstantin was talking about focus. You need to have a clarity about what you want to show, what is really proving the case, and what do investors and pharma want to see. And then I think Eric was talking about the importance that you have investors on board that share with you that vision and are going to go to support you to get the critical data so that you really can come to conclusions. So that's more or less what you hear from the panel, but the panel is only part of the story. So if people want to share their view on the topic of innovation cycle and what it does to your stage of the company, that would be very welcome. But we don't want to force anybody, but if somebody wants to comment.
This is where an audience participation starts. Yeah. So any volunteers at the back row? Yes?
don't all at the
Yes.
same time.
Excellent.
Just
Thank you.
one
Thank
after
you.
the other, right?
Thank you.
Thanks for the interesting panel. So our lead asset is an immuno-oncology small molecule, and we're seeing there is less interest by VCs in the past 18 months to invest in this kind of thing. So I was interested in that approach to go to overseas markets and how you think the cycles may be aligned or maybe not aligned in the Asian markets and the American markets, or do you think it's a global thing? Because there was the example of someone going to LG Chem as their first investment. So do you see that as an option? Or, so what do you kind of see as the cycles in different geographies?
I think it's an excellent question, which perhaps complement by what were, for example, the requirements from your Korean investors. Did they require clinical data before they would go in? Or are some other Oriental investors less keen on clinical data? Would they be interested to support an immuno-oncology project at earlier stages? Shall we start with Eric?
So related to the modality small molecule, I didn't hear that it's not in the scope today of interest of investor, but maybe you are more updated on that. I know some very nice companies that are developing for interesting target in the tumor microenvironment, small molecules, and that raise a lot of money on the Nasdaq. So it's still feasible, even if the cycle is different now. You know, you need to see where you are in the cycle and when you will go up or down. But for the Asian, once again, Asian, it's an area where I knew there is a pool of investor, at least when I started the process 6 or 7 years ago, and we had attraction from this company LG Chem that was interested by cell and gene therapy, and they really were interested to have a cell line to have antigen presenting cells. You know, so it was exactly in the spot of the strategy of the company, and so the match was perfect. And we discussed and made the due diligence, presented. We had some clinical data already on melanoma, a small academic feasibility study, plus a lot of preclinical trial. And we succeed to get this deal, and it was the beginning of the Asian story of the company. You know, and based on that, I could build on having investor from the Asia. And when you have Asian investor in general, it's long-term investor. So at least it used to be in my experience, and when you have one of key opinion leader in terms of investor, in general you have a lot of followers, and sometimes you don't even see the followers, but they follow. You know, so it's very specific, but it's good in a way because you know, in my board I've just one representative of the 6 Asian investors, so it's completely manageable. One person talked after to the others, so it's something to try. But to answer specifically to your small molecule, maybe it's more Thomas that could answer if it's today of interest or not, or the other people. I don't know.
Anybody else have a comment on this topic?
Let me add one on this one.
Please.
But I think, and I'm intrigued what you just said, Eric. But it's not bad. And what you see is for entrepreneurs, but even probably more for investors. You have leaders and followers, right? Mm-hmm. So, I mean, who of you have heard, I'm very happy to invest in your company if you have a lead investor, right? Right? I mean, there are so many followers, you can't count them, right? The question is about who are now really the ones that you have to convince. And maybe that's just for the debate, one approach to consider who are really the parties, and I'm talking about pharma investors, who are ready to go into the risk, to go with you and take on some risk, right? So and maybe that's where you have to focus, right? Because you can spend a lot of time talking to the followers being in pharma, or you need to get to the people who shape the field, and you need to understand what is the dynamic. I mean, just because you asked the other's opinion, right? Maybe I'm just sharing. So but that's something maybe for discussion. Who has more followers than leaders for your company?
I think it's a very fair question, and of course, panel, our co-chair is allowed to have an opinion. Fortunately for this panel, but I think that there's a very valid point. You know, how do you identify which rock do you have to turn to find these leaders in risk-taking that follow your vision, understand what you're doing, and are not discouraged by sort of recent technical failures in a related field, and see, oh, wow, this is really exciting. How do you do that? I see you're keen to say something, please.
No, I was just listening very intently. No, I think it's a great point that Andreas made. I mean, I wonder what they're earning their management fee for, those VCs who only ever follow other VCs. I don't know. But
I can tell you they all think that they're leaders, right?
Yeah, I mean, I think the field's a little bit prone to oversimplification as well. I don't think necessarily always and in all circumstances are VCs and pharma in lockstep in terms of which therapeutic areas they're investing in or how they will invest. And I'm talking here about strategics versus institutional investors, for example. So I don't think necessarily, I wouldn't be deterred necessarily if you're having to speak to a lot of companies to find that one company who has that belief in what you're doing, because I think the hardest thing to bridge, in my experience anyway, is like an asymmetry of belief. So you've heard everybody on the panel here, we all believe in our companies and what we're doing. But if you're walking into a meeting and the person sat on the other side of the table just doesn't believe in immuno-oncology, right, that they've taken a view in general, that's a very hard place to come back from in that context. And I don't know if the panel's got any view on how you overcome those sorts of barriers. So I think it is about finding people who fundamentally share your reason to believe in the area that you're working in, are willing to do the work, take their own view and lead, be that a pharma investor or a venture investor, and build from there. But don't waste time and energy trying to bridge an asymmetry of belief, which I think is extremely hard to do.
Sometimes fundamentalism is very difficult to overcome. How about that, Aram?
Yeah, I think it's a good point, and from the context of a company working in glioblastoma, I think there's a few observations I'll make there. It's much easier to talk to investors who you know are interested in the disease. And because many times with glioblastoma, you know they realize, oh gosh, it's glioblastoma, then forget it, because I don't even want to talk to you about that field. But we're discovering investors who are very interested. This is more, I'm finding a US style of investing where you have people who have a personal interest in this. There's a venture capitalist in the US whose wife sadly got glioblastoma, but he's now become one of the biggest advocates. He's pulled RA Capital into the field. He's connected with the Brain Tumor Society in the US, who also acts as kind of a hub for who's interested in funding glioblastoma research. So those are the types of places that I think are types of places you need to go to when you're in an indication that's perceived as very difficult and very tough for funding. And I'll make another comment about platforms, because we're an RNA aptamer company. And some of you may remember Macugen, which was an anti-VEGF RNA aptamer that got completely destroyed by the Roche antibodies. So I think the RNA aptamer field really looked horrible until last year, when if we talk about leaders and followers and being willing to take early risks, you saw Astellas wait until 2 months before approval of an RNA aptamer for geographic atrophy, but then they bought them for 5.9 billion. So I think it's that I hope will change the perception of RNA aptamers in the field a little bit, when people are only thinking of Macugen from 20 years ago.
There are certainly many different technologies that come and have their day. I mean, some of us remember Albert's selective toxicity textbook on how do you kill cancer. Well, if cells eat faster, so you just feed them poison. And if cancer grows faster than the other cells, well, it dies faster. Now, from that going to targeted therapies and then kinase inhibitors, great, kinase inhibitors are fantastic. And then you start getting resistance. Oh, kinase inhibitors are not so good. You have to make another one. And now we have a very creative kinase inhibitor. It happens to be in immuno-oncology modulator, right? Apollon. That is bringing something that is kind of like, oh, well, not another one, into, well, this is quite an interesting one. Can you tell us a little bit about that?
Yeah, I mean, happy to do so. I think in the end, it's not about the others, it's about us, right? So we have to make the case, and we have to really make the case and ask ourselves, is the things that we are doing within our company, is this something of relevance, having seen others failing, and why am I different, right? I mean, blaming others is easy and say, oh, they are not interested, but you need to create the interest, and you need to be, differentiated enough to say, we believe that this is a good case because X, right? We have learned from the failures of others because Y. And now, look, I mean, as you were saying, Esteban, I mean, having a kinase inhibitor and call it an immuno-oncology drug is appearing strange. But of course, there are intracellular targets that play a role in immune suppression and play a role in immune activation. And why not? And if this small molecule has an effect on the immune activation, also has an effect on top on the tumor cell directly, that is a wonderful case to have. And if you can combine these effects without having any tolerability issues, that is, I think, a perfect match. You combine actually two arms, right? You combine the immuno-oncology with a direct targeted therapy, and you combine it into one drug. That's perfect, right? And we have other examples in the company where we say, hey, there's one biological driver that's an immunosuppressive receptor that drives innate immunity or inhibits innate immunity. And there's another key player that drives adaptive immunity. Can we make a more sophisticated molecule that tackles both of them simultaneously? Can we do this? And that brings you to the next level. And then you can say, look, hey, we are combining two biologies, and therefore we believe we have a higher chance than having the one biology only. So it's about us, streamlining ourselves, have a clear strategy, understand where others are failing, understand where the gap is, and streamline your program, your company, into exactly this direction. And that's this consolidation is not coming as a surprise. For me, it's not. Remember, we had more than 1,000 active clinical trials combining X with PD-1. And why should this be successful? And that was simply naive. It was naive. And now you see, okay, there's a consolidation. But after the consolidation, what happens? The winners will survive, and it's about us to say we want to be the winners and streamline our programs accordingly.
It is a question of understanding the biology. I mean, people were trying this 1,000 clinical trials sounds like high throughput clinical screening, which is obviously an absurd concept now that in retrospect, but really understanding of the biology gives you idea that, well, we might use an old tool, even if it's an aptamer, come and do new biology using an old tool that was a solution to a different problem, or taking the dendritic cell line and say, look, by the way, you can teach a dendritic cell line to dance in a particular way, and cancer vaccines were out of fashion, but this one works. And so this is really a question of identifying the gap and trying to push through. But there's always going to be a time from the moment you identify the gap until you come with the solution. And that requires belief from yourself, of course, and also from the investors and ways to support. Is there something that you would recommend in that case? Maybe Konstantin, what do you feel is a way in which, from identifying the gap to getting the results, how do you bridge the people in the faith?
I think there's one word that probably works best, and I would call it quality. Quality in the data package that you start and that you prepare right from start. Because at the end of the day, the better and the more diligent you are right from experiment number one, I think the better and the clearer you are to you, and you can be true to yourself throughout the program. Because there's so many programs that are being rushed into clinic, and in clinic you fail, and afterwards there's actually you consider half a year of why did it fail. And there's many, many times that you come up with things that, I should have known and have looked at that. And that goes back to understanding the target and understanding the modality you utilize for that specific target. So I think, honestly, from day one, if you're very diligent, if you're very true to yourself, and you really do high-quality experiments, I think then you just de-risk your asset through all kind of stages of development. And that's preclinical, that's discovery, and that's clinical. And of course, we've heard in the oncology setting and in the PD setting today, not everything is translatable into clinic. We know that, but I think exactly because we know that, we can do the best to identify the right model and not just use any model to say, this is great, looks data, and this is why everybody should buy the program. It's not going to be, because people are smart and they will know it's the right model or not.
Excellent. If I synthesize what you were saying, Konstantin, with Apollon, I agree, and at the same time, I would add it's very important that you can build your story and communicate all of this, right? I mean, basically, that's of course the typical researcher trend, that you are trusting your own data, get convinced about your own thing, but you create something like a story, a perception around your company, which is, sounds a little bit marketing wise, and I'm an R&D guy, but fundamentally there is your company is going with a notion which is very intimately linked to the story that you are being able to tell, which includes all the aspects that Apollon was talking about, to be really differentiated, not just one order of many. So that's what we then see at the investor side. There are some companies who are fantastically suited to tell a story, and they may have the same data as another company who just is not telling the story compelling, right? So, but we have another 2 minutes to go, so that's the last chance for the absolute urgent question that somebody wanted to raise.
I might have missed this part of the discussion a little bit later, but were there any thoughts about PROTACs in oncology and the progress of this modality in the last year?
We have not mentioned the protein degraders, but just provocatively, agonists and antagonists are just as good.
But PROTACs is at the moment en vogue, right? So
exactly. Well, you know what? I was just trying to be slightly provocative in the sense that you can get a lot of biology that is trying to be replicated in a difficult way with the PROTACs by classical pharmacology. But let's put that to the question. Really, the question is really is something en vogue better just because it's en vogue, or is there something really a great opportunity there for novel pharmacology, novel biology, and novel therapeutics? And that is a fundamental question.
But the answer is differentiated, and I like PROTACs, right? But I think at the moment it's probably easier to come with a PROTAC story because that attracts attention than coming with another story, right? So I think that, I mean, you may have missed part of the debate, but that's where we're debating about these cycles of what's hot, what is less hot, and how you can win, sticking to your concept and idea. And unless there is no other burning last question, I try to finish this more or less on time. Thank the panelists, their courage, their entrepreneurship, and thanks for the discussion. And I hope the audience enjoyed what we were debating here. Thank you so much.
Thank you.